Hello?
Hi.
Hi, how are you? Hello, Abhishek? Hello, Kush? We'll wait a couple minutes for people to come into the meeting. We had a time change in North America, so we're going to wait. Hopefully people can make this time. Hello, Tharun. Oh, left. We have two chat. Okay, hello, yes. Okay. Well, I guess, I don't know, like I said, there was a time change in North America, and I sent out reminders, but I don't know what people got and what they didn't. So welcome to the meeting. I guess I can start now. We had some things planned from some of the people who are working on GSoC, but I don't know if when they're gonna be here. So we'll start and we'll go over regular meeting business, and then we'll get into the see who shows up into the meeting. So this is our DevoWorm meeting, and usually it's more populated than this by different people from who've been here a while. So, and we do record these on YouTube. So I'm recording them for people who couldn't make the meeting, so they could see some of the issues that we have ongoing. I'm going to go over some issues today. I'll talk a little bit about GSoC, as I normally do during this time of year, and then I want to actually start though, but I want to ask people if they want to introduce themselves. Looks like there's
Hi,
Shruti. Hello, how are you?
I'm good. How are you?
Okay. So yeah, could some of the new people? We have several new people here. Oh, here's Susan. Hello, Susan.
Hey, Susan.
It looks like she's got a new setup or something.
Yeah. We can't hear Susan.
Oh, Susan, yeah, you're muted, I think. Anyway, yeah, there she is. But I can't hear her. Can't hear you. Okay, hello. Yeah, I can't hear you.
Oh my god. She's looking so cute.
Yeah. Yeah, it's not coming through. I don't know. Here's Mayukh. Hello, Mayukh.
Hi, Bradley.
Hi, how are you? So yeah, I was just saying that we were gonna. I wanted to start by having the new people introduce themselves. We have Param and Kush, and I don't know if we have anyone else. So if you want to use the chat, if you can't speak, that's fine. But I want to get a sense. Param, why don't you go first? Introduce yourself.
Hello everyone, I'm not sure if
Yeah, you can't really hear you very well. Okay, we have in the chat, we have hello, yes, hello, Param. Yeah. So, if Param is going to type his answer, we could actually hear from maybe Abhishek. He wants to introduce himself.
Hello, yeah, my name is Abhishek and I am currently a second year student pursuing engineering physics major.
Okay. And what are you interested in specifically in terms of the GSoC projects?
I am currently looking for working in deep learning, but my basic interest is in computer vision.
Okay, good. Do you have a project that you're thinking about contributing to or applying to?
Yeah, this one, this meeting is for that project, 3.1 updating DevoWorm.
Okay, that sounds good. Thank you. What about Kush? Do you have introduction?
Yeah, hi, I'm Kush. I'm also a sophomore from Mumbai, and so I found this project very interesting as I find it's like a nice introduction to machine learning and computer vision as a whole. So as a whole, regarding GSoC, I'm not very sure if I'll be contributing purely through the GSoC because I don't believe I have the skills required for some of the projects, but I do think like this is a nice organization that maybe I can contribute to in the future and maybe hang out with people and understand some skills regarding that.
Okay,
I hope it's okay.
Oh no, you were gonna say something else?
No, I was not going to say something else. I was
Okay.
just asking if that is okay, as like if I'm trying to be a rather long-term contributor.
Oh yeah, that's great. Yeah, that would be good. Yeah, we talk about GSoC, but it's not. It's pretty competitive. So I mean, there are always opportunities to contribute through the project, and then maybe make that into something a little bit more substantial. So it's always
Yeah.
good to Well, nice to hear from you. Welcome. We also have Param. Did you just want to type in your introduction into the chat?
Yeah, is it better now?
Oh yeah, I can hear you now. It's better.
Yeah. Hi everyone, I'm Param. In general, I just look to contribute and have fun. I've not really decided projects, etc. I do find some interesting, but let's see how it goes.
Okay. Yeah, that's great. Yeah, thanks for just checking it out and seeing what's going on. So this, each meeting is not necessarily representative of everything that's going on. We just cover different topics as they come up, and we can certainly talk about things if you're interested in them from the meeting, or if you find something that you might be interested in. We can talk about it and put it on the schedule and talk about it in more depth. So it's definitely, there's a lot going on in the group. We have a number of long-term members, and then we also have people who come in every once in a while and check in. So I would get in touch. I'll show you in a minute. We're building an onboarding guide, so we'll talk about that in a little bit. But I wanted to talk. I wanted to ask people. I know there have been a number of people in the Slack this week who have been, kind of wanting to show off some of the stuff that they've been doing with respect to some of the GSoC projects. They've been working on little tutorials, or they've been working on little, trying out different things. So does anyone want to present anything?
I was looking forward to presenting the cellular automata thing that we were talking about last week. I did some research about it. I'm still into it. And I won't say I know everything about it as of yet. But yeah, I would like to present after everyone is, all the people are done, if there's anyone.
Okay, yeah, that's good. We will do that. But I know people had like some notebooks or something that they had that they were quickly working on. And is anyone prepared to do that this week? I know that I think Yash maybe, or
Yes, yeah.
yeah.
I implemented a Jupyter notebook based on fuzzy classic CVs algorithm.
Okay.
Can I present my screen?
Yeah. And then I think we'll go to Mainak after that, and then to Shruti.
Can you? Yeah, is my voice coming? I don't know.
Yeah, you can hear you. Yeah.
Okay.
All right. So Yash.
Yes, I'm presenting.
Okay.
Is it visible?
Yes. Can see your screen.
Yeah, it's visible.
Okay. First thing is for configuration, drive configuration.
Okay. All right. So could you walk us through it?
Sorry?
Oh, could you walk us through it? Explain what you did here? And,
Yeah.
Okay.
The first thing is for mounting drive. Like I have to give access for my drive for database.
Yeah. I think your audio is cutting out. Yeah.
These are some
Okay.
images from.
Okay, so
Like
those are
we
the
are
original.
using this frame 1662 copy path and
Yash, am I audible?
yes.
Yeah, could you please just fast forward it to the algorithm itself? Just like we wanted to know how exactly it works. So that
Okay.
we
First
don't
I
like
loaded the images.
we can if the outputs are already visible, there's no need to run it again. We can just straight go to the code itself.
Yeah.
Okay.
Just give a high level overview of what it is.
Yeah, sure. So here I have defined number of cluster key is equal to 2. The first cluster is from the Basileia and the second one was the background. Then these are the compute centroid, distance matrix, update member and update member function. So all these functions are working like the fuzzy seaming clustering algorithm. First we have to define membership matrix.
Oh, right. You told me about this. So this is the fuzzy approach. Yeah.
Seaming clustering
Okay.
algorithm. So here membership function. Here member we have initialized membership function like number of Dirichlet equation, random initialization Dirichlet.
Okay.
These are shown in here. I have prepared it.
So this is fuzzy C means. Yeah.
This is the main algorithm. So UIJ is the membership matrix and VIJ is the central.
Okay.
So we are passing FCM function, image and initialize member function and number of iteration that we want to refer up. So what it will do is we compute all the distance from the center and the center are update accordingly every each iteration. Like first we have initialized the member value, then we find the cluster center by this formula by calculating the distance and then we update member. Then we again calculate cluster center, then we update membership function. We repeat all the iteration till we get epsilon difference between the cluster center like epsilon. That was that we defined with that. We can iterate through as many of time as want. Like here, I try to do this 5 iterations. That will do fast. After iteration,
Okay.
that will take some. First, I am showing the first one is this one is the image that we get from our database like this. This was the original image. The second one is the we have the second one is the membership matrix. This one
Okay.
we found from this. The third one we thresholding this like this numpy.var member. Like if the value of pixel is greater than 0.5, we give 0.00255. That means we give the pixel 1 value, and if the less than 0.5 pixel member value, then we give it a 0. By masking the third image and first image, we get the final result is like green and green. Okay.
Okay.
Okay, yes,
Here we have implemented only 5 iterations. Yes.
yeah, I'm sure the results will be much. Yeah, please keep sharing your screen if possible. Let's just put
Yeah,
the
yeah.
So, yeah. So I was saying that like the results were really good when you ran it for a lot of iterations. I know it's like just 5 iterations, so it's kind of not good right now. But the point here is like as far as I saw, the whole thing is implemented in NumPy. And
Yes.
when you're running NumPy, which means it means that you're running on the CPU. So
CPU, yes.
it's pretty easy to just port this port this whole code to Torch and move it to the GPU. It will be
Yes,
way
yes.
faster.
And
I suggest you to do that. That would be really good.
that part can be implemented in this. I have simply used the for loop and its complexity is
It can be vectorized.
Yes.
Instead of like using 3 for loops on top of each other, you can just vectorize the whole thing. And maybe use Torch and move the whole thing to the GPU. It will be like 10 times faster at least.
Actually, currently I am working on the PyTorch. I am familiar only with TensorFlow.
Okay,
So
so
I will
Torch is just like NumPy.
Yes.
You can basically replace NP with Torch and most of it will work. Like Torch.zeros is the same as NP.zeros.
Yeah. I will do it in 2-3 days, and I'll share it with you.
Okay.
Yeah, sure.
Well, that sounds good. Yeah. Thank you, Mayukh, for your advice on that. That sounds like a good idea. And again, this is an opportunity for people to get feedback and show what they're doing. I think it's good. Next up, we have who wants to go next? We had hello, Krishna. I think was it my
Yeah, I can go next.
Yeah.
If that's okay.
Yeah.
So I'll just share my screen real quick. So just hold on.
Okay.
Okay, so can someone tell me if you can see my screen or not?
Not yet. Oh, there we go. Okay.
Not yet.
All right.
Okay, so I'll begin the presentation. So my presentation is basically about the work that I've been doing since the last week. So it's kind of like a summary of my work. So is the slide visible?
Loading.
Okay.
Okay, there we go.
So it's visible now, right?
Yes.
Okay, so I'll start. So my presentation would be about exploring the spatial distribution of cells in the C. elegans embryo using 3D interactive plots. So I'll go ahead to the next slide. Okay, so the dataset that we are dealing with is basically the EPIC dataset, which is really popular. So the data is like it looks somewhat like this. So it has many columns. Like it has this column called cell, which basically says the cell.
Mainak? Like we lost him for a minute here. Well, we have some things in the chat here. So yeah. Okay. Oh. Yeah, I don't know. Mainak. Oh, hi. Hello, Akshay. Welcome. I don't know if Mainak knows he's not on here. I don't know why that cut out, but let's see. Are you there, Mainak? Why don't we? Why doesn't Shruti go first, or Shruti go next, and then Mainak? We can come back to Mainak when he's back up and running. Oh, there he is. Let's see. Yeah, he's frozen again. I don't know why.
Yeah. So is my voice coming?
Yeah, now you're okay. You cut off for a bit, and you lost your screen.
So that was an issue with my internet. So I'll just share my screen again.
Okay. Yeah. So
let me know if it's coming.
Okay.
Okay. So is it visible?
Not yet.
Okay.
There we go.
So,
Okay, we got it.
yeah, okay. So I'll go ahead to the slide that I was on. So this is the data set that we're dealing with. I guess a lot of you might be knowing this data set. It's the EPIC data set. So it's basically a time series data set that deals with it basically deals with the embryogenesis of the C. elegans embryo, and it has all these columns, and it basically represents the embryogenesis process where we have all these cell lineages that gradually arise from just one cell. So this is the data set that we're dealing with. So I'll go ahead to the next slide. Okay, so our objective is not to deal with the other columns. Our objective actually just revolves around these columns. Like we got the cell time column, we got the cell column, we got the time column, and the XYZ coordinates of those cell lineages. So I'll go ahead to the next slide. Okay, so using matplotlib we can generate the positions of these cells from we can generate the path of the position of these cells as they progress from one point to another, and using this library we can just generate the static plots, and it's actually quite difficult to visualize the progress of time through these static plots. And like if we have to visualize time, then we'll have to save each plot separately and then create a GIF later on, which is very tedious, and there's actually no way to quickly find the coordinates of each point. Suppose like you want to find the coordinates of this corner point, so it's almost impossible to do that without any specific code. So I'll go ahead to the next slide.
Yeah.
Okay, so this is where these interactive plots come in. So using these interactive plots, like as you can see, so the GIFs are visible, right?
Yes, you can see them.
Okay, so I'll just share the link to the GIFs so that you guys can I'll just share the link real quick, because I don't think it's sharing smoothly in the screen share, right?
Okay. Okay. Yeah, there we go.
So I'll just resume the presentation now.
All right.
Okay, so it's still loading, right?
Yeah, you can see it.
Yeah. Okay. So as you can see in these interactive plots, we can basically move the cursor and we can change the perspective, and we can zoom in or zoom out, and so this gives us a much better insights. This gives us actually a much better insight of what we are dealing with. Like in the previous slide, we had the cell lineage DPA. So the path is traced here using this blue line. So, but there's no way to change the perspective or to zoom in or out or to find the coordinates. But in this case, you can just zoom in and check the coordinates of each point just by using the cursor. So I'll go ahead to the next slide. Okay. So as I was speaking, so as I had told already that the dataset that we're dealing with, it has it is basically a time series data. So visualizing the time is really important in this case. So using these interactive plots, we can also visualize the time. So as you can see, there's a scrubber. So you can scrub through the time points, and you can stop at any point of time and change the perspective, zoom in, and find the coordinates of each cell lineage. And yeah, another thing that I would like to say is that the time ticks here, they are not in the correct order. That's actually an error in my code, so I'll be fixing that soon. So these two movies, they basically show the capabilities of these plots. So there's a stop button and there's a play button, so you can just play to let to basically see the whole process unfold in front of you, and you can pause at any time and check these coordinates, and you can zoom in or zoom out to analyze further. So I'll go ahead to the next slide. Okay, so to summarize this, for these kinds of data like 3D time series data, it's very difficult to gain insights from just static plots. So that's why I decided to experiment with these interactive plots. So these plots, they actually enable you to change the perspective, to zoom in or out, to project the coordinates, to visualize the movement direction. Like as you can see in the plot to the left, there's no way to know that the cell has went from this point to this point or from the lower part to the upper part. But in this case, I've actually color coded each point according to its time point. So you can clearly see that the direction of movement of the cell with time. So that's something that's somewhat of an advantage in this case. So I'll go back to the next slide. Okay, so this is something that I actually plan to do. I don't think I'll be able to do it until at least a month from now. But this is something that I got planned. So my plan is to basically take these segmentation maps from the DevoWorm model outputs and stitch them together in form of a 3D closed surface segmentation map, because these frames, they basically represent many slides of the same cell. So they could be in theory stitched together to form an interactive plot that could be rotated, zoomed in, or maybe even like you can even find each coordinate of each point using the cursor itself. I guess that would be way easier for the scientist or the researcher to find insights from the given segmentation map, rather than having these 2D slices of, rather than having these just 2D images. So this is what I plan. So I guess that's it for my presentation, and I would really like some feedback from you guys right now. So thank you.
Thank you. Let's keep the slides up. First of all, does anyone have any questions?
Yeah, I had a few questions.
Okay.
Mayna, could you just go back a few, like the slide in which you were showing the interactive plots?
Yeah. So is it this one?
No, the next one.
Okay, hold on. Okay, is it this one?
Yeah. So these interactive slides, like, may I know in which platform are you making these?
I'm actually making these
I mean,
in
which
Python.
language? Okay.
I'm
They are
actually
made
making
in
these in
Python,
Python.
which is
Yeah, this is Python.
interesting. But
Yeah, and
my question was,
okay, go on.
wait. Okay, my question was that basically we have talked about interactive demos in the project proposal. So the thing is it possible to integrate these interactive plots in some kind of a web backend, in a web page?
Absolutely. Like you can, unlike the Matplotlib plots which store PNG files, unlike them, you can just like these plots, you can just take the whole thing and you can just embed them inside websites. So it will remain as interactive as here in the website itself. So that would be a great addition.
We won't need any sort of code to get these kind of plots, right? So is it possible to somehow use this data and like directly make a port where we can where the user just throws in the data and he gets this kind of an interactive plot? Is it possible?
Yeah, absolutely. Like I believe in the future, like it is absolutely possible that the user will just copy paste the link of the CSV that he has, and he can generate these plots. So that's entirely possible.
Okay, interesting. Yeah, thanks.
Yeah.
So any other questions, anyone?
Well, I had some comments. So I wanted to go over, like, this is good work, very good work. I wanted to go over some of the aspects of the data set. Could you go up to the first, let's see, slide 3 maybe. Maybe slide 4. When you say that there's a lineage and you say DPA, what are you measuring? Are you measuring just the position of DPA, or like DPA and all the cells that come before DPA? What is that?
This is actually the cell lineage. This path actually shows the path followed by the cell of the lineage DPA. So that's what this is dealing with.
Okay, so then that would be DPA, DP, D, and then the precursors. So it would be
Yeah. So this DP actually, this string, this name actually comes from the cell column. So this data is what I'm showing here. This is the exact data. Like this name was present in the cell column, so it could be Z3 or it could be any other cell lineage. So that's what.
Okay, so when you say lineage, you mean kind of like everything that led up to that cell.
No. I say that exact cell, that like the cell of that lineage.
Oh, well that's
So
a single cell, yeah.
Yeah, so I guess DPA does not include DP and D itself. It's just DPA.
Yeah, exactly.
So, and that kind of brings me to another point, which is the data here are not normalized, and this is not like necessarily have an impact on the visualizations, but like one way we have to deal with the epic data set is we have to normalize the data somehow, the position data, because they're kind of raw positions, and if you take like a bunch of them from different embryos, they're not all they're in a similar frame, but you can't you can't say like each point is equivalent. So like they'll track cells for a number of embryos, and then there has to be a way to normalize across the different embryos that were sampled to get numbers that are comparable. So we've used different strategies for that, like pre-processing the data. So you could do like an average position. You could start like from a center position for each embryo and just say, there's a center point 0, 0, and then there's like a maybe like a z-score, a plus or minus away from that center point, and then that gives you like a framework to work with so that you can make comparisons. So this plot here would be like the cell as it's dividing, it's kind of moving because they do sample it at different times. But I mean, this is good work, but I don't know what the if we play around with the data and normalize the data a little bit more, and maybe look at like how cells go from like as they divide where they move to. That might be interesting as well.
Yeah, so you're speaking about the normalization part, but like as we are just dealing with the XYZ coordinates of the data, so like I saw the features of these columns, and I really didn't think that it needs a normalization because they're really quite inside limits. So as we are just dealing with a limited set of columns, so I didn't really consider normalizing them.
Hello. I think that is right. Normalization is important, especially when we are dealing with not just a single cell but with multiple cells.
Yeah.
When we are scaling up, normalization would be important. Like a mean of 0 and a standard deviation of 1 would be really nice.
Yeah.
Absolutely. And that's something that when we use different data sets, we don't really have a mechanism for that with our. I mean, some of the data that we have that's been processed in DevoZoo, for example, has that property where we've normalized it. But like the raw data sets, we don't really have community standards for, is what I'm saying. And I don't know, maybe that's something that we can develop as people start to use the DevoLearn platform more and start getting into some of the other data sets. Thinking about strategies of how to normalize the data, because it depends on the data set that you have. If you have something that's been acquired, very rarely are data acquired very uniformly. A lot of times they'll acquire data, they'll get like samples, they'll measure a sample, and they'll not get the exact interval that the other sample was sampled at. So, there's a lot of that sort of improvisation that goes on in data analysis, and it's sometimes people think it's science, it has to be all systematic, which is what we're trying to do, but often when you collect data it's not as clear cut. You have to kind of make some decisions about it. So that's very good work. Thank you, Mainak.
Yeah, that's great work.
Yeah. And I look forward to this. There's actually a paper where they're doing something similar to this concept image.
Yeah, exactly. In fact, I was actually I took this concept art from that paper itself. I guess it's in your, it's in the Google Drive folder, right?
Yes, it is. Yeah.
Yeah. So in fact, I took this. I took this illustration from that paper itself, just to show how it might look like. So,
oh, okay. Yeah. Good. It was a paper on 4D embryo modeling. So they took the 3 dimensions of space and then the 1 dimension of time, and then they created like a virtual model of the cells, and that that was what they were doing. But we could do this in a number of different ways. That would be so that's one way to do it. But we're trying to go for actually a 4-dimensional representation, because you have the 3 dimensions of space plus the 1 dimension of time. But and then there are other things, like we can look at the angle at which a cell moves and it divides. So when a cell is a single cell and it divides into two cells, and then they either stay next to one another, or what more likely happens is they shift around in the embryo. And so we can measure that, use that as like a fifth parameter, which is like angle. So we can go on like, in a number of different parameters that we can use to characterize the embryo, not just space. But that's a good start. Thank you.
So I guess Mayukh wanted to add something, right?
No, I didn't want to add anything else, but I really look forward to integrating these kind of interactive plots into some web-based, like some website or something like that.
Yeah, that would be good if we had like a way to integrate it into the web. Ideally it would be like a resource where someone could punch a button and get a visualization. But I don't know how hard that would probably be. Very hard to do, especially if you have you'd have to have your data set pretty defined, which is part of the part that we don't really talk about. We talk about the machine learning part, but then you have to host your data like we saw in the last demo. You have your Colab notebook, and then you have underneath your Colab notebook you have a data set somewhere that you're drawing from. And so that's another part that we have to kind of figure out, or that isn't like just trivial. But so yes, thank you for that.
Yeah, absolutely, and thank you all for the comments. So yeah, I'll stop sharing my screen.
Yeah. And then now Shruti, if she's ready.
Yeah, I can present.
Okay.
So, what I'm going to talk about today is cellular automata, and this is something that I and Bradley have been talking about some time back. So, first of all, what cellular automata actually does is creating the simplest possible complex system ever, and it's a very absurd line, but let's see how we're making a complex system the simplest one. So this is something what cellular automata looks like. These are different cellular automatas which are made from different rules that we'll see in a moment how they are made. So first of all, cellular automata is nothing but they are computational models that are represented by grids like you can see here. This is a 1D. I'm giving you an example for 1D cellular automata just for understanding purpose. So it's like an array or a list, whatever you want to call it. And what we do is we concatenate and we just populate the list and put some numbers, zeros or ones, because we want to keep it simple. And so we saw there are some grids, then there's something which is called as the cell state, and with the neighbors. Neighbors, for example, for one are these two, the one before it and the one after it. So these are the three main things that are required to let the automata work. So this is something which this automata could do if we use the rules properly. But how is all this movement happening? Although this one is for a 2D automata, but let's just go and find out. So the first step, like I said, generation 0, generation 1, generation 2, first of all, these are nothing but time steps at time t equal to 0, t equal to 1 or t equal to 2. So what we have is we have this first random number or some sequence which we have, and now we want to predict what it would be in the next time steps, like generation 1, generation 2, and for that we can see that the cell state at time t, if we suppose the first one, would be depending upon the neighbor at time t minus 1. So what this actually means is, suppose we want to find out what would be the value of here, what value here, we would have to consider the element itself in that position, one before it and one after it. So for computing that value, like 0 here, how did it come? We have certain set of rules. And if we have different set of rules, there are many set of rules, 256 actually, I'll let you know in the next slide although, based on which we can find out the value of the next automata, for the next generation step for the automata, and then for the second generation, third generation, and so on. Also, the 0 here, for the animation, like I showed you here, all the zeros are representing the white color which we see, and the ones are representing the black color. So once we do this step, many number of times. This is a pattern, a very beautiful pattern which we will get, and it was not something we expected out of just numbers like zeros and ones. This is called as Sierpinski's triangle, and this triangle was particularly found out using rule number 90. It's called rule number 90 because when you decode this, when you convert this binary string into a decimal string, it comes out to be 90. So this particular example was for rule number 90, and we have 256 such examples, such rule sets possible. And some of them give very interesting outputs, some don't, some are very dizzy, and you feel like you'll just go, you'll have some headache or something out of it. So out of all these 256 set rules, only a handful produce some compelling outputs. Like this one, which you're seeing, is not a very, something which you will find very different or attractive. And this is yet another example of one of the rules. So what is the use? This is something which actually, like Stephen Wolfram, the person who's really into this, and there's a lot of work he has done in this field, he said that the basic aim of using these automatas is to represent how life, how we can represent life instead of using mathematical equations, how we can represent life in the form of patterns and symbols and in a very different way. And you can see the applications of these. For example, the rule number 90, which I showed you a few slides back, can be clearly seen in this shell. And there are many more examples, like examples that we have in biology. We have many examples of this. This is a visualization of the lattice cat automation, which is also made using the computational cellular automata. And there are there are so many patterns in the world. Like, for example, whenever some metal breaks or some material breaks, how does it form a particular shape? That can be determined using this. Or what are the different patterns we see in environment, different biological patterns that we see. All these can be really nicely understood using cellular automata. So it's basically the main goal of cellular automata is stimulating the natural world with code. Instead of like how all these patterns can, because code is what is there. We want to have a solution for everything, and for that we use code. So cellular automata is an approach, or it is a step in the direction of using code for stimulating all the natural world things, and it is quite proficient in it. So the next thing, this is what I have done the last week, and after next week probably I'll be doing I'll be working on the game of life, and also the main aim of using cellular automata, as I and Bradley were discussing, how we can use cellular automata to upgrade our model, because we have already been using deep learning and all these approaches, and their optimization of course and go hand in hand, but using a different approach for dealing with the data that we have and the model we have would be really great. So yeah, this is what I've done right now. So
that's very good. Thank you. Are there any questions?
Yeah, the presentation was really nice. That's the first thing that I would like to say. And the second thing is that, Shruti, you are speaking about simulating the natural world with cellular automata. So another thing that I actually found interesting way back, I guess a couple of months ago, when I was reading about cellular automata, is that you could also simulate computers inside cellular automata. Like a person named Paul Rendell, he had simulated a Turing machine inside John Bomber's game of life. So
I read about it.
Everything actually. So yeah, that's something that I wanted to add. So like,
in fact, sorry.
Okay. So these seemingly simple systems, they basically can lead to extremely complex behaviors. That's the interesting part in it. And like if it's possible to simulate a Turing machine, then I believe we can also simulate whole computers in that, like 64-bit machines.
That's
something really interesting there. So yeah, the presentation was nice. Yeah, that's all.
Yeah. Thank you.
In fact, there's one more thing. Stephen Wolfram has given out 3 questions regarding this only. In fact, particularly for Rule 30. And each question is worth of probably $10,000. So anyone interested in wanting to earn something can go and check out those questions. They are really they are about the randomness. Like, how can you determine? Like the one I showed you for Rule 30. there's no particular pattern. So he wants to know that is there a possibility that the randomness of the rule would either be repeated or there won't be any repetitiveness, or they're very nice questions, and they're something that you think about once you read them. You'll be forced to think about it more and more. So yeah, if anyone's interested to earn $30,000, can look at them.
Very good. I had a question about you showed seashell in the presentation, and you said that you can simulate the surface, which is true. We were, earlier, maybe about 2 years ago, we had been talking about doing, taking seashells and putting them on a mount and then taking images of the surface, rotating it on like a turntable, taking images of the surface, and then assembling like a 360-degree movie of that surface, or like some sort of flatten out all those images so that you have like this sort of one-dimensional or two-dimensional sheet of that pattern. And so this would be collected from actual seashells. So I was thinking about, it never really got off the ground, but I was thinking of the parallel between that idea and like simulating it in a cellular automata, and getting a sense of like pattern formation and those sorts of things. Yeah.
We can look into that. Actually, this is something new and interesting that I found out, and I would be going through it more and letting you all know what I'm doing. So let's see where this goes.
Yes. And for those of you who are new to the group, we do a lot of we like to explore pattern formation because we're interested in development. We're interested in pattern formation, and especially in cells and in life. So you saw before, you have you have these 3D models of the embryo where you have different patterns forming. You have cells moving to different places, and sometimes you have on like things like seashells, you have surface patterns of different chemicals that get deposited, and we would like to understand that process. So I think this is definitely a part of this. We did a couple years ago, we did something, we had a platform that we used, we have a paper on something called Morphozoic, and I don't know if Shruti read that or saw that paper.
Yeah, actually I was looking for the paper. I think I misplaced the link of what I was thinking of. Thank you for reminding me that I need that link, and I was not able to find it.
Oh, well, I'll send you the link of the paper, and then there's actually a GitHub repo as well where it's housed. And then, that's one version of it, but, you might find it interesting to play with. So, yeah, we'll be continuing this discussion in the coming weeks. Yeah. Let's check the chat. Okay. Susan says, place the shell on my new microscope. Yeah, you could do that. We were thinking of putting it on like a turntable and having it just rotate at a certain speed, but if you could get everything simultaneously,
last week we saw Susan's microscope. It was interesting. I wish I could use it. Susan, we can't hear you, Susan.
Oh, okay. No. She's going to put it in the chat. So, yeah, the Turing machine made using Conway's Game of Life. This is the link that Mainak shared. Susan says, send me your samples. So,
okay.
Yeah.
Sure, I was actually looking for something, using it myself. I really doing stuff with things, so yeah. It's okay.
Mayukh said cellular automata in 3D space would be interesting. So people have done this. It's a little harder to implement, but people have done like the 3-dimensional case where you have cubes instead of cells, like the 2-dimensional cells or 1-dimensional. So it's harder to implement, but it's people do it. Yeah. And then finally, Mayukh has some news here. DevoLearn 0.3.0 has 112 weekly downloads on February 2021. So this is the project 3.1. This is the software that Mayukh published last year, and we've been working on version updates on this. And so we had 112 weekly downloads. So it seems like people are using it, or at least interested in it. So congratulations on that. Yes, this contains the stats for DevoLearn. Okay, so this link here, snyk.io advisor. Okay, so you can go to that link if you're interested in seeing those. I'll look at those later. So let's move on here. We'll go over past the top of the hour. If you have to leave, that's fine, but I'm going to go on maybe about 20 minutes after the hour on some various things that I wanted to cover. So we have some deadlines, just to remind you. We have deadlines coming up for various submissions. So if you're interested in contributing to a submission, let me know, and I can point you to the right place. We have this list of submissions, things that have been submitted, things that are in the process of being submitted, and things that are farther out. A couple of things that are new this week is that I submitted let's see if it's on here. Yeah, I submitted a abstract to the INCF Neuroinformatics Assembly, and this is an overview of DevoLearn. So this is similar to the DevoLearn Flash Talk given at the OSF Virtual Conference for Online Education. And we'll be going through those slides in the coming weeks. And the reason this is important is because INCF also sponsors our Google Summer of Code, and so it would be nice to get that out in front of all of INCF to show them the progress we're making on that. Krishna Katyal and myself and Jesse Parent submitted a paper on a comparison between artificial neural networks and biological neural networks, and that was just submitted last night. So congratulations. This is something that's actually a full paper, a conference paper, so we don't know yet if it's going to be accepted, but it should be actually out on the archive pretty soon. So it'll be on. I'll send that out in the channel. And then NeurIPS is coming up, so there's a paper submission deadline for NeurIPS. You have to submit the abstract by May 19th and the full paper by May 26th, and the paper details are on the website. It's like an 8-page double-column format paper. So if people want to submit a paper that they are working on, that's fine. It's very competitive, of course, but you can always try. And so that's for NeurIPS. There's also, I don't know if this CompleNet paper will happen, but I targeted this thing called Euler Paths for Life, which I've talked about in previous meetings. Also, this growth form and theory of deep learning item, which is currently an abstract, and I have a number of people on it, and I also had Daniela Cialfi, who's in the Slack. She wants to contribute to this too, so we're probably going to work on this, maybe fleshing this out into a paper or a longer document. Right now it's like 2 pages with references, and the idea here is to talk about like the morphogenetic patterns like we saw with cellular automata, with some of the stuff with the microscopy analysis, but to think about this in terms of like the theory of deep learning. Like, why is it that deep learning networks can maybe generate some of these things or inform us about some of these things? And so I don't really know where this is going to go, but we'll maybe start working on this a little bit more and flesh it out. So those are all opportunities to contribute to the group. So if you're interested, let me know. If you have things that you think would be interesting to submit to, let me know, and that would be good. Also, I wanted to pull up the Slack. I wanted to make sure everyone knew where we were with the Slack. What we have here is if you come into the Slack, if you're invited in or you join, you're going to join. You're going to want to join DevoWorm as the channel for this group. You'll probably also want to join some of the other channels. There are other projects like C302 and a couple other projects. You'll see the channel list. But I would recommend, if you're interested in working in this group, joining 3 channels, maybe 2 channels, depending on your interests. So one would be the DevoWorm group, and this is the channel. And you can see it's, we talk about the meetings. We have people joining in, and people post like things like this is Yash's work that he presented on today. He just kind of put a pointer in this channel for us to pay attention to it and give further instruction, which is what this channel is for. This channel DevoWorm is sort of like for everything. But if you're really interested in like the DevoLearn platform and the DevoWorm Learn software and machine learning more specifically, then you have joined the DevoLearn channel. And that's this channel where we have different things. I have this agent-based modeling package that I found. I put a link to it here. I CC'd Shruti on it, so she might find that interesting. I've put in links to different things, so like for some of the people coming in, I kind of point them to different resources, and Mayukh is pointing them to different resources in here as well. Courses that might be of interest. And then we have GSoC 2021. So if you're specifically interested in GSoC 2021, then this is a channel you can join. And this is kind of like a more general sort of conversation about the different projects. So, we don't want to like fill up DevoLearn with things about GSoC specifically. We want to keep this for kind of like the machine learning computational stuff. DevoWorm, we want to like make it very general, but also have a lot of biology in here. And then GSoC 2021 would be very specific to GSoC projects and Q&A related to that. So that's our Slack. And I wanted to also talk about this onboarding manual. So this is something that I'm trying to find it here. Okay, here it is. So this is the onboarding guide. So this is something that I'm going to be filling out here. Thank you to Mayukh here for putting together all of this. These are more details on the specific projects. So we have each project. We have some references listed here. So you have, their papers for 3.3. There's some papers you need to read. For 3.2, there's some data sets that you can link to. And there are all sorts of things that you would need to get started on these projects. We'll be updating this regularly, but I wanted to put it in GitHub so that we could all access this. These are the slides that Krishna came up with, and I wanted these slides in here because I wanted to point people to things like understanding what the relationship between INCF, DevoWorm Group, and OpenWorm Foundation are. So INCF sponsors the projects. OpenWorm is sort of our home organization, and then DevoWorm is our research group. And so these things will maybe be a little bit helpful in the application process. One thing we do need to add to this is a section on how to create a timeline for your project. So I'm planning on putting that together soon too, and I hope to have this ready by the beginning of the application period. So I know that and I'll organize it so that people can like index it so that people can go to a certain point in the application process if they're interested in a specific part of the onboarding guide. So that's what we're so if you're I think I put it in the channel, but if you're interested in DevoWorm, or if you're interested in GSoC, you're interested in contributing, you should look this over. But even if you're just interested in contributing to DevoWorm in general, you might want to take a look at this. And I'm going to maybe add in some more general DevoWorm things in this onboarding guide as well. So that's all that. If you have questions, let me know about that. I don't know if we have anything in the chat. We have two things in the chat. Okay. Oh, Dick is here. Hello, Dick. He's a beginner in Mathematica. Anyone prepared to answer simple questions? Okay, so yeah, he's going to contact Shruti. So one of the things we do in the meetings is go over some papers. And so the last maybe 20 minutes, I'll go over some papers. And for some of the new people, don't let this be too intimidating. We talk about developmental papers, and I think it helps people understand the development side quite a bit more. So today I'm going to talk about a couple of new papers, and I think Dick has sent me some papers this week, and so thank you for that. And so let's see. First paper is this. This is Lab on a Chip. This is a magazine on the biological magazine. They're interested in working with like microfluidics and other types of things where you can have what they call a lab on a chip. This is a C. elegans animation here where they have it on a tabletop, or I guess a lab on a chip, and they're doing these different experiments with it. These are eggs that C. elegans lays. It's a stylized cartoon, so they're showing like this egg laying process. And I think the idea here is electric egg laying, so they have like the dipole here with the electricity flowing across it, and that's supposed to be this electric egg laying. So electric egg laying, a new approach for regulating C. elegans egg laying behavior in a microchannel using electric fields. So this is so C. elegans, they lay eggs, and they lay sometimes several hundred eggs throughout their lifetime, and the eggs kind of are derived from the germline, which forms actually very early in development. And so they form these form these eggs, which are basically germline cells, and then they start laying them as they become an adult. And so this is a paper on talking about the effects of electric fields on adult C. elegans egg laying. So they're using this microchannel, which is a specific type of instrument that they use. So they do this in a microchannel. It's discovered and correlated with neural and muscular activities. So basically they're delivering an electric field. They're putting the C. elegans in this microchannel, and they're affecting the egg laying capacity. And egg laying, of course, involves neuromuscular behavior. They have to actually, take the eggs out of their egg laying tract, and it has to go out into the environment. So they have to squeeze the eggs out, but they don't squeeze them all out at once. They lay them in rounds, and so this is what they're trying to facilitate. So the quantitative effects of worm aging and EF strength, direction, and exposure duration on egg laying are studied using egg count, body length, head movement, and transient neuronal activity readouts. So they're looking at like the number of eggs laid. They're looking at the properties of the worm, and then they're looking at the neural activity of the worm in the muscular activity and the electrical activity there. Electric egg laying rate increases significantly when worms face the anode, which is one pole of the dipole, and the response is EF dependent. And then longer EF exposure results in shorter egg laying responses during shorter egg laying response duration. So basically they can control the duration of egg laying with this electric field. Worm aging significantly deteriorates the electric egg laying behavior with an 88% decrease in the egg count from day 1 to day 4 post-young adult stage. So they lay their eggs in rounds, and sometimes, they can be pretty old when they keep laying eggs, but there's a certain window within their adult life that they can lay eggs. Worm aging significantly deteriorates the electric egg laying behavior. Yeah, we got that. Fluorescent imaging of intracellular calcium dynamics in the main parts of the egg laying neural circuit demonstrates the involvement and sensitivity of serotonergic hermaphrodite-specific neurons. So these are all neurons that are involved in egg laying that they're identifying. Then there's a mutation that also results in the reduced rate of egg laying, allowing the use of this technique for cellular screening and mapping the neural basis of electrosensation in C. elegans. So they actually say that C. elegans can sense electric fields, and they're demonstrating that with this technique. This novel assay can be parallelized and performed in a high throughput manner, so they can do a lot of C. elegans at once and identify some of these behaviors. So this is a good. I'm gonna put a link to this folder in the channel, and so if you want access to this, I can also put it in the in the Slack channel. Okay, so Mainak gave the links to his presentation slides here, and Krishna's email is here, and Shruti's email is here. Okay, so the next paper is the segmentation clock paper. And this is a new window to the segmentation clock. So this is a paper on what they call the segmentation clock. So what is that? So the abstract of this paper is segmental organization of the vertebrate body plan is established by something called the segmentation clock. And so this is a molecular oscillator that controls the timing and periodicity of somite formation. So you have these cells that are formed and there's a certain periodicity to it. Given the dynamic nature of the segmentation clock, in vivo studies in invertebrate embryos pose technical challenges. So vertebrate embryos are like fishes, humans, mice, etc. As an alternative, simpler models of the segmentation clock based on primary explants and pluripotent stem cells have recently been developed. So these are different experimental models that we use to understand some of these processes. So we use C. elegans as one model organism, but we also use things like explants and pluripotent stem cells, which are cell models, cellular-based models of these processes. It's much easier to study these complex biological processes in a model system, and so we use these types of model systems. They enable more quantitative analysis of the oscillatory properties and expand the experimental repertoire of the segmentation clock. So we can study more things using these models. Crucially, by eliminating the need for model organisms, so they don't use model organisms, they're using these cell models. In vitro models allow us to study the segmentation clock in new species, including our own. The human oscillator was recently recapitulated or replicated using induced pluripotent stem cells, providing a window into human development. So we can actually take like our own body's cells, like skin cells, and turn them into stem cells using a certain molecular process. And I can talk about this in an upcoming meeting, but suffice it to say that this is a common model of sort of these types of processes. Certainly a combination of in vivo and in vitro work holds the most promising potential to unravel the mechanisms behind vertebrate segmentation. So let me see if they have any images in here. It shows a little bit more about what they're doing. So if people don't know, in development we have this thing called segmentation. So an embryo is this mass of cells. It starts out as this mass of cells, and then you get differentiation into different cell types. But you actually also get organization based on location. So this is why we're measuring position of cells. We're interested in the sort of the organization of the embryo. But in other organisms other than C. elegans, there's also this pattern formation by like segmentation. So you have like different parts of the embryo that become different sections of the organism, like the tail or the head or central parts. And so there are different model organisms where you can study this, but it's very hard to study because you have to have a very specific setup experimentally. And so this is a way to get around this experimental setup to do this in cell models. So they're actually looking at different molecular markers here, and they're looking at segmentation and the spatial patterns in the embryo and in the developing organism. So this is an example from like gastroloids. We've talked about these before. These are these groups of cells that kind of that people grow in a culture, like in a bioreactor to sort of develop these multi-cell bodies that exhibit these developmental properties. So they differentiate and partially differentiate and show all these interesting properties. 3D differentiation, which is where they use a three-dimensional culture, and then 2D differentiation, where they use a standard cell culture. So I'm not going to go through the whole paper, but this is just a preview. So if you're interested, you can read about it. So I also have three other papers, which I won't talk about, but I wanted to kind of like set up the theme here. So this is a paper on where the part mirrors the whole, interactions beyond simple location. And this is a paper about emergence in development and how we might understand the difference between different locations in an embryo or in a system more generally, and then how those interactions sort of allow us to see emergent phenomena. So we looked at the cellular automata. We know the cell and its location isn't the whole story. We have these patterns that emerge across the whole grid that we have in our cellular automata. In this case, you'll have emergent properties in an embryo that emerge from the interactions between individual cells. And so they talk about this, because this idea of emergence is very hard to get your head around and to use in like quantitative measures. So they're actually looking here more at the sort of the philosophy of this and some of the other types of things that are considered. So this goes back to the reductionism versus emergentism debate. So reductionism is where we want to look at the things that are the easiest to sort of isolate and understand in their isolation, like a specific gene or a specific cell. And emergentist means we want to understand what the cells are doing collectively, or the genes are doing collectively. And so they lay out a lot of this. So if you're interested in like the philosophy of science, this is a good paper to look at. Then there's this paper, Common Principles of Early Mammalian Embryo Self-Organization. This is a recent paper in development. They talk about these common principles of mammalian embryo self-organization. So C. elegans aren't mammals. They're much simpler than mammals, because mammals actually have a lot of, they don't have this sort of these programmed cell lineages. They have a lot of intercellular signaling, which determines the fate of the cell. And so they talk about the common principles in this type of embryo. And so the abstract reads, pre-implantation mammalian development unites extreme plasticity with a robust outcome. The formation of a blastocyst, an organized multi-layered structure ready for implantation. The process of blastocyst formation is one of the best known examples of self-organization. The first three cell lineages in mammalian development specify and arrange themselves during the morphogenic process based on these interactions. So despite decades of research, we haven't really defined any principles for this. In this paper, though, they talk about the role of physical forces, molecular and cellular mechanisms, and how those drive self-organization and the formation of these lineages. And so, again, the lineages are just basically going from a single cell to multiple cells and looking at cell divisions in that process and tracking it and saying, is there something about the cell lineage that's important? And so this is how we kind of get in mammals. It's a little bit complicated because you can't really reconstruct a cell lineage deterministically. You have to observe the cell lineage, and there's a lot of signaling. But this paper goes into a lot of this literature, and it's probably pretty hard to read for someone who's not familiar with development. But I just like to point out that there are three things here that we talk about in the meeting quite often: physical forces, molecular mechanisms, and cellular mechanisms. So that's something you can follow up on if you want to. Finally, there's this 4 simple rules that are sufficient to generate the mammalian blastocyst, and this is related to the other paper. And that if you read the other paper, you should also read this paper. And I'll go through the abstract really quickly here. Early mammalian development is both highly regulative and self-organizing, meaning that there's this regulation between cells. So cell-cell interactions are determining what happens in the embryo. and self-organizing, meaning that the interactions between cells are really determining what goes on. It involves the interplay of cell position, predetermined gene regulatory networks, and environmental interactions to generate the physical arrangement of blastocysts with this timing mechanism. So, things have to come together to form this blastocyst. So the physical arrangement is determined by all these factors here. However, this process occurs in the absence of maternal information and the presence of transcriptional stochasticity, which means like when genes are expressed, they're expressed in a way that's not deterministic. They're kind of expressed probabilistically. And stochasticity here does not mean the same thing it means in machine learning, just so you know. How does the pre-implantation embryo ensure robust reproducible development in this context? And so the question is, what are the principles here? In this paper, then, they talk about it utilizes a versatile toolbox that includes complex intracellular networks coupled to cell-cell communication, segregation by differential adhesion, which means the cells are adhering to things differently, and apoptosis, which is cell death, programmed cell death. And so these are all different factors that they point to. Here we ask whether a minimal set of developmental rules based on this toolbox is sufficient, and to what extent these rules can explain mutant and experimental phenotypes. So whether you have mutants where they have a gene knocked out and they have different phenotypes from what they call the wild type, or what that means. And so they look at a bunch of things here. They build a quantitative model. They build this so they're actually building. They're putting all of these factors into a developmental set of developmental rules, and then they're modeling using an agent-based model of physically interacting cells. And so from this quantitative model, they find that it reproduces specific phenotypes and provides an explanation for the emergence of what we see in real embryos. And so they also talk about some of the molecular mechanisms. We conclude that a minimal set of rules enables the embryo to experiment with stochastic gene expression and could provide the robustness necessary for evolutionary diversification of a gene regulatory network. And so that's a lot of jargon in there, but this is basically the idea. So you have a single cell, you start to see cell division, you see this first rule implemented, polarity, so cells sort of form sort of basic polarity between each other. Then you have this signal, this molecular signaling, which guides the cells in different directions. You have differential adhesions, so these cells are adhering to one end of the embryo. And then apoptosis, where some of these cells die off purposefully to shape the embryo. And so this is how they implement these rules, and they observe it here in this I think this mouse embryo here. So they're doing this. They've built a model. Then they build this in silico model, or this computational model, where they actually look at the physical interactions between the cells. So they're looking at these different cells, different cell positions, and they're assigning different physical forces to them. And then they're looking at the model with different parameters. So a potential versus distance plot here, where they're showing differences between what they call the trophectoderm and the inner cell mass. So this is the inner cell mass, this is the trophectoderm. And so they're actually looking at these different parts of the embryo and how this model sort of models it. So then they have these different logical rules that they implement. And so this is actually quite relevant to, it's sort of adjacent to the cellular automata work, in that it's more of it's not really a free-form model like cellular automata, but it also relies on some of the sort of logic of a cellular automata. So implementing rules, having cells that are influenced by its neighbors, and so on. So I think that's enough for the papers for today. And let me look at my chat here. Let's see if we have anything. So Dick says, let's read his latest paper here. And we have Susan, molecular signaling may be due to forces on cells. That's true. If molecular signaling is influenced by environmental forces. One of those are forces on the cells themselves. Dick says, are we on the cusp of a new paradigm for biology? So this is his paper that he just published in Biosystems. And then he says, embryo arises from the inner cell mass. So that inner cell mass that we saw is where the embryo, a lot of the differentiating part of the embryo arises from. And then inner cell mass is exposed to fracking, which is a paper that Susan presented on a while back, talking about like hydraulic pressures between cells in an embryo. So that's all I have for today. Thank you everyone for attending those who stuck it out through the papers. Thank you especially. Did we have any comments or questions before we end the meeting? What is environment? That's a good question. It could be like just anything outside of the cell or outside of the biology. But that's a good question. What that means. I mean, people use that term pretty freely, but it's not. I guess you could define it, but it's basically anything outside the cell or outside the embryo or organism, I guess. What is information content of the environment? That's another good question. That's something that depends on how you operationalize it, I guess. If it's like something that we can detect through information theory, or just something we say is like information, which is often what people do in some of these papers. They just say, well, there's this, we introduce a perturbation, that's information. We don't know that for sure, really. I mean, that's my answer. There anything? So I just wanted to follow one last thing. So we have this in the next couple weeks. We're going to have people who are, trying to contribute to GSoC or coming into the organization due to GSoC, and so let's make sure that we get, we kind of keep on top. I'll keep on top of that, and Mayukh will keep on top of that. But if you have any specific questions, don't hesitate to ask us. Dick asked, embryos for axolotls grown in distilled water at constant temperature. What does environment do? I mean, I don't know. I guess if you varied the temperature, it would be that would actually be information content. But I'm not sure. I mean, it's an excuse for WTL or WTF. Yeah. Yeah. So. All right. So any other questions? If not, we can probably end the meeting. Yes, thank you. Nice to see everyone, and if you have any questions, we'll be in Slack or email. And so thank you for joining us at the new time. We had a time change in North America once again. So everyone outside of North America, it's going to be a time change maybe for the next couple weeks. I don't know how your time changes go, but we had our daylight savings time. So thanks for attending. Have a good week. Thank you.
Bye.
Bye.
Bye.